Hello and welcome to this new Clinical OMICs webinar sponsored by Genomics entitled Optical Mapping in Rare Genetic Disease Diagnosis. I'm Jeff Bogaliskas, Technical Editor for Clinical OMICs, and I'll be your host for today's presentation. As a class, rare diseases constitute a large number of clinical cases, each representing a unique set of diagnostic challenges. Interestingly, however, rare genetic disorders often result in structural genomic variations. While short-read sequencing approaches can detect deletions, they are less useful in identifying and localizing insertions in the genome. Additionally, when structural variations are large or in complex regions, even long-read sequencing approaches are typically not able to characterize them properly. Thankfully, researchers have an extremely efficient methodology in optical mapping to identify large structural variations, determine their breakpoints, and position large insertions uniquely in the genome. Clinical researchers are finding that optical mapping can be applied successfully in the molecular diagnosis of rare genetic disorders through the use of techniques such as fine karyotyping, copy number analysis, and breakpoint analysis of microdeletion syndromes. In today's webinar, we're going to find out more about the advantages of optical mapping and how next-gen sequencing hasn't quite lived up to the promise of identifying large structural variations that result in rare genetic disorders. Now it's my pleasure to introduce our speaker for today's event. Dr. Pui-Yan Kwok is a distinguished professor of human genetics at UCSF, where his research focuses on the development of new approaches toward whole genome analysis and their application to gene mapping and haplotyping. Dr. Kwok has pioneered the high throughput of discovery of SNPs across the human genome. He was a member of the International HapMap Consortium Steering Committee and has authored over 110 publications along with 3 patents. Today, Dr. Kwok will demonstrate how optical mapping has consistently proven to be an accurate and efficient way to identify large structural variations and how it can be utilized effectively in rare disease research efforts. But before we begin, I want to remind the audience, as always, to stick around for the Q&A session that's going to occur right after Dr. Kwok's talk. Don't be shy. Send in any question you have at any time during the presentation. Just click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. We'll try to get to as many of your questions as possible. Okay, now I'll turn the floor over to Dr. Kwok.
Hello, everyone. Today, I want to share with you our work on using optical mapping in rare genetic disease diagnosis. A rare genetic disorder is defined as a condition with an incidence of less than 1 in 2,000. However, there are more than 7,000 rare genetic disorders, so in aggregate, it affects 25 million people in the United States, which is about 7.5% of the population. Most of the disorders are familial, meaning they run in families. Now, when a mutation in one gene is necessary to cause a condition, it is an autosomal dominant condition or an X-linked recessive condition in a male. When it requires 2 mutations to cause the disease, then it's usually an autosomal recessive condition or X-linked recessive in a female. However, some cases are due to de novo mutations, and they're usually found in the conditions where only 1 mutation is required. So, as a dermatologist, I want to share a few examples with you. So many of these diseases are caused by mutations in one gene. For example, in this case, this little child has no tyrosinase because the tyrosinase gene is defective. And because of that, the person cannot make any pigment. So he has albinism. So it doesn't matter what happens, he has no hair color, no iris color, and he is kind of devoid of pigment. In another condition called xeroderma pigmentosum, this person is highly sensitive to UV irradiation because he lacks the gene to do nucleotide excision repair, so that when UV light damages the DNA, he cannot repair it, and so he is very prone to skin cancer and premature aging. Now, before the Genome Project, to identify the molecular defect of rare genetic disorders is an arduous process. In a process called positional cloning, for example, in this case, this person has fragile skin in a condition called epidermolysis bullosa simplex, and the Weber-Cockayne variety. In order to study the genetics of this condition, one has to assemble a huge pedigree so that we can identify the people with the disease versus the people without the disease. So this is an autosomal dominant case. Then we use genetic mapping to narrow down the region where the defective gene is residing. Then we clone the region into small pieces, do Sanger sequencing, and identify the mutation. But everything changed 20 years ago when the Human Genome Reference became available. Now we know pretty much all the genes in the genome. So instead of having to do huge pedigree analysis, we can focus on the patient individually. And this was greatly aided by two developments. One was a team at the Broad Institute came up with a way to massively produce oligonucleotides and then put them on beads so that one can then capture pieces of DNA from the genome. So this is the bead-based target capture ability. So a few months after this was published, a group at University of Washington used this technology to look at individual patients with rare genetic disorders and identify the gene involved. So basically, with the cost of sequencing getting lower and lower for short-read sequencing, one can then use either capture sequencing or whole genome sequencing to then align the reads to the reference and identify pathogenic variants that explain the phenotype. So the trade-off then is that instead of using a pedigree analysis to use genetic to identify the location of the gene, now you just sequence all the genes and pick out all the variants and then try to figure out which variant is involved. So these are what we call experiments of one. Now you basically not use any genetic information. You're just using genomic information to identify the pathogenic variant. However, this approach has some limitations. There are some technical limitations. For example, for exome sequencing, the coverage could be uneven because if you capture the target sequences unevenly, some of the genes will not be interrogated properly. Even when you do whole genome sequencing, there are limitations based on short-read sequencing. For example, if the regions are duplicated, or there are structural variations, or there are sequences not found in the reference genome, then the whole genome sequencing will not be able to figure out whether there are variations involved. And then, of course, for short-read sequencing, there is no facing information, which means that if you have a compound heterozygous situation where one gene has two mutations that are deemed to be pathogenic, but you don't know whether the two mutations are on the same chromosome or on two chromosomes. In the former case, the patient still has one normal copy of the gene and will not be affected in a recessive situation. In the second case, because they are compound heads, they will be affected. So due to these limitations, the current success rate of short-read sequencing in rare disease diagnosis is about 30%. If you're really good at pre-screening the patients, you can get the percentage a little better, but for all comers, the current success rate is only 30%. So the diagnostic failure is actually due to two things. One is what I just mentioned. The short-read sequencing does not give a complete representation of the genome. But there's another problem, and that is because we have incomplete biological knowledge, which limits our ability to identify the causal variant among the many variants in each person. And in fact, for each of us, we carry several pathogenic variants in our genes. It's just that we're not affected by it. So going back to the first situation, the fact that we're not looking at all the variations in the genome when we use short-read sequencing alone. So one big category is structural variations. Structural variations arise during recombination when our germ cells are being formed. So when we form egg or sperm cells, the parental chromosomes come together and then they have a crossover and recombine. In effect, they're shuffling our parents' genetic information when we form our cells in eggs or sperm. So in most cases, the recombination occur in a balanced way so that nothing happens. However, we have a lot of repetitive sequences in our genome so that when they are misaligned during this recombination process, then the resulted daughter chromosomes will either have a deletion or duplication. And sometimes the recombination happens on the same chromosome. Then you can come up with inversions. And at times, the recombination is between two different chromosomes, and then you get translocations. So here are some examples of the different kinds of structural variations. We have deletions, we have novel sequence insertions, and then we also have these mobile element insertions, such as alus and line elements. And then there are times when we have duplications, and the duplications can be in tandem, meaning the two copies are right next to each other. And they could also be interspersed, meaning the new copy landed somewhere else. And we already talked about inversions and translocations. So in a survey of many people, one identified that there are about 1,000 structural variations, meaning bigger pieces of DNA that is placed in different locations in our genome when we compare it to other people. So there are about 1,000 of these in each of us. And if you look at the population as a whole, there are thousands of them. And in fact, this study was done 10 years ago with maybe a few thousand people. And now that we have a lot more information, this number will continue to go up. Now, short-read sequencing cannot handle large structural variations because the read length is only about 150 base pairs, and then the pieces of DNA that we do paired-end sequencing with only expands about 300 to 400 bases. So with large structural variations, basically the pieces just cannot be put back together when they're repetitive elements. So this was partially solved by using array CGH, comparative genomic hybridization. Basically, you tile a whole bunch of probes across the genome on an array, and then you label a reference DNA with the sample DNA with different dye colors, and then you mix them together and hybridize on the array. So when the probe is present for both of these samples, then you get a yellow color. So that means that there's no difference between the reference and the sample. However, if the sample has an extra copy, as in the case of a duplication, then there is an excess color from the sample versus the reference. So in this case, it's green. On the other hand, if there is a deletion in the sample, then the reference color shows up. So in this case, is a deletion. So using this approach, people have been very successful in identifying big structural variations in genetic diagnosis. But there are some limitations. One of them is that you cannot tile enough probes on the array so that the resolution is actually about 300 kb. Anything smaller than 300 kb, the clinical labs usually will not call because the sparse placement of the oligos. But there are other limitations. So if you look at a deletion situation where the control has this piece, but the patient doesn't, and then when you look at the array CGH signal, there's a depression here. So that's no problem. However, if you have a duplication, now you have an extra piece in the patient. Here, you see a signal. So you know that there is an extra copy in the patient. However, you don't know where the new copy is found because there is another situation where you have an interspersed duplication where the new copy is somewhere else. You still get the same signal, which means that there is no information on the location of the extra copy. In the case of an inversion, you have the piece of DNA in a different orientation between the control and the patient. Here, there is no signal at all. You just don't know because there is no copy difference. And so you cannot detect anything when there is an inversion. Likewise, you cannot see anything when there's a translocation, especially a balanced translocation, because part of the DNA stays with the original chromosome, but the other part of the DNA has now gone to a different chromosome. But because there is no copy number change, you get the same signal. So once again, not detected. So because of these limitations, we have been thinking about how do we get the most information about variation from the patient in rare disease genetic diagnosis. So we came up with an idea to combine both whole genome optical mapping and long-read sequencing to build genome assemblies. And if we have built these assemblies, then it's free of the need for a reference if we look at the parents and the child as a trio. So when we do this, we can actually detect everything, small variants, structural variants, and with sequencing added to mapping, we can actually have breakpoint resolution and also with haplotype resolved phasing of the assemblies. So we have done just that, and we have actually developed an automated pipeline to identify the pathogenic single nucleotide variants, structural variants, and compound single nucleotide and structural variants. And this work is coming out in a few weeks in the MPJ Genomic Medicine study. Okay, so when we talk about this, we rely on two single molecule technologies. So the first one is using long molecules for mapping. So this replaces the old BAC and YAC cloning in the genome reference sequencing project. And then the second technology is to use medium-sized molecules for sequencing, long read sequencing. So the first is, of course, optical mapping. And the optical mapping on nanochannels, which is marketed by Bionano Genomics, is based on an assay that we developed in our lab. The original version is we're using nicking and filling with fluorescent nucleotides, and then using nanochannel analysis to determine the spacing between the labels. And with that, we can construct the genome maps. So this was published like 8 years ago. So basically, we label the DNA, stretch it out into nanochannels, and then after imaging and translating the image information into a linear molecule with labels at various places, then we can put the pieces back together into a consensus map, a contig. So this is very useful because if you look at the reference genome with the predicted nicking sites or labeling sites, you can compare your own sample and see what is the difference. And in this case, there is a tandem repeat found in the test sample in the blue versus the reference sample in the green. And so because we have single molecules spanning all the way across, then we know for sure that this tandem repeat is real. And we can also identify large insertions where it's not found in the reference genome in the green. In our sample, we definitely have that. So this is a pretty big insertion, but because, once again, we have big long molecules coursing completely across it, then we know that this is, in fact, the case. And inversions are particularly easy to find because we can see the patterns of the reference on both sides of the inversion, which correlates really well with our test sample. But then in the middle, the pattern basically flipped around. So with the long molecules crossing the entire region, we know for sure that the inversion is there. So when we use this approach and we actually look at the general population, we found that there is quite a bit of missing content in the human genome reference. So we published on this last year, and we found that there are, 2.6 gigabase of the genome covered by most people. But there are 51 complex regions that are bigger than 200 kb each, spanning a total of about 60 megabase that are very, very complex, and there is no one reference that can really give it justice. And as I said, we found about 50 megabase of new genetic content not found in the reference genome. So this is quite important because we have a paper coming out soon that tells you that some of this missing content are actually quite common in the general population. It's just that the donors that gave us the human genome reference do not have it. And some of these are actually expressed in genes and so forth. Okay, so that's with optical mapping. With the medium-sized sequencing for long read, we have used the 10x Genomics linked read sequencing approach. But as you know, this is no longer available in the marketplace. And so you can use other long read sequencing approaches for this. And with the linked read sequencing, we can use the barcoding to help us place the short read sequences into larger contigs. So by combining the optical mapping and the linked read sequences, we can actually place a lot of the almost all the short read sequences into a big, long hybrid map assembly. And in fact, we were able to reconstruct a human genome really well. So the theoretical number of maps, because of the chromosomes that we have, is 46. We were pretty much down to that, and with phasing of much of the genome. So now I want to share with you the different things that we can do with optical mapping. So the first is to look at these really highly complex regions of the genome, especially the segmental duplications. Although they are only 5% of the genome, they have very, very high sequence identity and very low copy number. And they are hotspots for recombination. And so quite a few microdeletion syndromes are due to these segmental duplications. The famous ones are, for example, the DeJour syndrome, the Williams syndrome, etc. And so we have looked at several of these regions and identified the different patterns of duplications and inversions and so forth. Here's an example. So this is the 16p12 low copy repeat region. So you can see that this is the region here. And in there are several duplicates. So the same color tells you that these are highly identical sequences. So for example, A here in the orange is about 69 kb, and then so forth. So this region is highly complex because when we look at the different individuals, we see very different patterns. And here we can show you that in the different populations, Africans, Americans, and East Asians, Europeans, and so forth. And so we can see that they have very different patterns. And in fact, there are dozens of patterns for each of these cases. So using this approach, we were able to identify and catalog the different haplotypes for these special regions. And then with that, we can look at patients with microdeletions. So here we have 4 examples. 2 came from chromosome 7, and you can see that because we have the map cataloged already, we can tell from the patient that the deletion has to occur between these 2 highly repetitive duplicates in the blue. And for this person, is the magenta duplicate. So the other 2 are found in chromosome 15. Once again, you can see the deletion pattern really simply, and then chromosome 16. And another example is from the 22q11 region for the DeJour syndrome. So here is a very complex region. We can actually see that the deletion happens in this shared region like this color here. And then we can pinpoint where the deletion is. And here are some molecules that span across this deletion point, so it can help us figure out that the deletion indeed came from this chromosome based on their patterns. So this is a really easy approach for deletion mapping of microdeletion syndromes. But if we add the sequencing to it, then we can do even better. So we just finished a case study using 50 patients with congenital anomalies. We use a de novo sequence assembly, combining optical mapping with long-read sequencing to look at the patient and the parents. Using an automated analysis pipeline, we identified the different scenarios. So with this, we were able to achieve 40% diagnosis of patients that are previously negative with whole exome sequencing. And then we have another 36% with strong candidates that our biological knowledge now, we cannot call it because of the ACMG criteria. However, we will do additional studies to validate these as real diagnoses. So I'll show you a few examples with using the remaining time how this can happen. So the first family is a 9-month-old female with craniosynostosis and syndactyly. And we identified a 32 kb heterozygous de novo duplication within the gene NHEJ1. So here's the BioNano optical mapping result. So here's the reference genome, and clearly in our patient, one of her chromosomes has a 32 kb insertion right here. And you can see how the markers line up on both sides. And so we know for sure that there's a duplication because the pattern here is repeated. When we look at the linked read sequencing, piling up the barcodes, we can see that there's a region where the proband has, high density of barcodes not found in the parents. So when we look at the sequences, we identified the two haplotypes by long-read sequencing, and we see that there's a duplicated region here based on coverage. And we actually resolved the breakpoints using both the mapping and the sequencing to this area. And basically, it duplicated part of this NHEJ1 gene. And it turns out that this duplication does not affect this gene, but the duplication now gives us an extra copy of an enhancer, a long-range enhancer in this gene that causes problems with the Indian hedgehog gene just outside this gene. And so it turns out that this is known. There is a syndrome called a chromosome 2q35 syndrome, where there are several families with duplications of this gene that turns out that there is an enhancer that causes a problem with the Indian hedgehog. And so this case is solved, and despite the fact that short-read sequencing didn't give us anything. And we know that it's de novo, so it's helpful for the parents to know that it will not happen again if they decide to have another child. So the second family is an 8-year-old girl with multiple congenital anomalies and global developmental delay and seizures. And for her, we found a bigger de novo microduplication, a 303 kb heterozygous situation on chromosome 4p16.3. So the BioNano optical mapping data, once again, shows that there is a big insertion here that is not found elsewhere. And in this particular case, there was microarray information, but it turns out that this was not called because it's in the borderline, about 300 kb. But when we look at this case, using linked read sequencing, we were able to identify this, once again, duplicated region not found in the parents, and we were able to resolve the breakpoint. And so this region turned out to harbor many, many genes, and it's also published that people with this kind of duplication have this very similar kind of phenotype. So once again, this is the de novo change. We were able to identify that, and the parents now know that they don't have to worry about the next child. And the third case is a 2-year-old boy with neuroblastoma and developmental delay. Microarray analysis of this child shows a lot of homozygous regions, meaning the parents are somewhat related, and exome sequencing not diagnostic. When we looked at the optical mapping data, it's clear to us that there is a de novo translocation between chromosome 1 and 9. So here you can see that there is a contact here from the patient that tracks from chromosome 1 to chromosome 9. And then the reciprocal case is where there is another contact that tracks chromosome 9 and then crossing over to chromosome 1. And so if you put these two contacts, line it up against chromosome 1, you can see that definitely they match up at the breakpoint. And you can see the same thing when you line up these contacts against chromosome 9. When we looked at the link-read sequencing data, once again, the proband has it, but the parents do not. And the breakpoints can be identified with sequencing data. We know exactly where it happens on chromosome 1 and chromosome 9. And the breakpoints actually disrupt two genes, AGBL4 and LINGO2. And these two genes are actually known to be associated with neuroblastoma. So now we, knowing that there's a translocation, we actually confirmed it with karyotyping. And definitely you can see the change here. And so we were able to tell the parents that it's an unlucky break that they have a child with this translocation, but it's not going to happen when they have subsequent pregnancies. So the fourth case is a little bit more complicated. So this is a child with clinical features of Cornelia de Lange syndrome, which has a lot of facial and kind of structural features and also developmental delay. So the doctors decided that, well, a lot of these cases have chromosomal changes, so they decided to do a karyotype. So the karyotype showed that there are three kind of chromosomes due to translocations. Chromosome 2,5, chromosomes 5,6, and chromosome 2,6. And so that's all they got, and they couldn't figure out whether these were the causes of the syndrome. So we looked at the BioNano data. So we found that there is a chromosome 2, chromosome 5 translocation, and a chromosome 5 and chromosome 6 translocation. And then there's another translocation that is likely an artifact because it's not seen in the karyotype and also is found in both the child and the two parents. So we're ignoring this one. But we couldn't find a chromosome 2 to chromosome 6 event. So because of that, we decided to really carefully look at the BioNano data. So it turns out that for the chromosome 5, translocated to chromosome 2 is due to this breakpoint at 36.9 megabase from the end of the chromosome. So you can see that really well. So chromosome 2 has this breakpoint. So in all directions we look, this is real. So we have this translocation. And then when we looked at the chromosome 5 translocation to chromosome 6. Once again, it checked out really well. But the interesting thing is that the breakpoint is at a different location. So instead of the previous one, where we have 36.9, this is at 39.5. So there is almost a 3 megabase difference between the breakpoints. So then, knowing this, we looked at chromosome 5 more carefully. So when we do that, we actually found out that there is a piece of, the chromosome 5 location breakpoints reside in a small piece of DNA. So between 5 and 2 here, and between 5 and 6 here. And so there is this almost 3 megabase piece of DNA from 5 that is now translocated between these two chromosomes. So putting them all together, it gave us this picture. So indeed, there is a translocation between 2 and 5 here, and translocation between 5 and 6 here. But then there's a other piece that is a translocation between 5 and 6 and 5 and 2. So basically, we can now explain the 3 chromosomes that are identified in the karyotyping, but in a more precise manner, knowing the different breakpoints. So in graphic form, this is what happened. So the 2 chromosomes, 5 and 6, recombined and exchanged material and gave you this situation. So with a balanced translocation this way. But in a second translocation between the newly formed 5-6 and chromosome 2, now we came up with this new situation. So in the end, you have 3 chromosomes in this pattern. And it turns out that the chromosome 5 region that got disrupted turns out to harbor this gene called NIPBL, which is the NIPBL cohesion loading factor that plays many roles in sister chromatid cohesion, chromosome condensation, and DNA repair. And it is well known that mutations in this gene result in Cornelia de Lange syndrome. So the final result then, that we can actually tell the people who did the karyotyping that if they can do a better, like using interface fish or something, they can actually identify this pattern, which is there's a translocation between 2 and 5, 2 and 5, 5 and 6, 5 and 6. So 2 translocation events with 4 breakpoints. So in conclusion then, I hope that I have been able to convince you that molecular diagnosis of rare genetic disease is challenging. And structural variations are responsible for some of the rare genetic diseases, that optical mapping is a very efficient way to search for large structural variations. And it's particularly useful in detecting insertions, inversions, and translocations. And if we combine optical mapping with long-read sequencing, we can conduct a full genome analysis that has the best yield in rare disease diagnosis. So I'll end it there. Thank you.
Dr. Kwok, excellent presentation with some really outstanding data. Thank you so much for sharing that with the Clinical OMICs audience. We really appreciate that. To those audience members who have sent in questions, thank you very much. To all of you thinking about sending in questions, don't hesitate. Send them in now. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. Okay, since we have a bunch of questions lined up and ready to go, let's get to the Q&A session. Bear with us for just a moment as we transition into the Q&A.
All right, everyone. Thanks for joining us for the Q&A. We have a bunch of questions for Pui. Pui, your first question, one of our audience members starts off by saying, you show this amazing diagnostic yield from the combinations of 10x and BioNano. If you use just each method on its own, do you know what the yield would be for each? I assume the variants were detected by both methods independently.
Yeah, that's a really good question. So to answer that question, you really have to think about what is the sweet spot for each of the technologies. So for 10x, basically it has barcoded linked reads, right? So it's very, very accurate because it's Illumina short read sequencing. But then you also get the kind of virtual long read from the barcodes. However, this kind of the context that you can build with that is still limited to maybe 100, 150 KB. So anything that is kind of small and anything kind of single base, 10x can detect very well. And so I would say anything that is smaller than a few KB to tens of KB, 10x will do just fine. But for anything larger, BioNano is the only way. And so as you can see in my examples, quite a few of the things that we found were of the larger variety. So I would say for BioNano, the yield will be really good for the large variations, but then everything else, 10x will do just fine. So it's kind of hard to, because we selected certain cases to work on. I don't know for all comers, I would say 10x will probably do maybe, no, for all comers without any pre-selection. Like we did a lot of exome negative cases, but if it's, all comers, I would say 10x will do maybe 40, 45%, and then BioNano can do similar, but all the big ones.
All right, great, thank you. Next question we have, one of our audience members asked, what characterizes the hotspots and why do they tend to break more easily?
Yeah, so hotspots are actually based on recombination during germ cell development. During recombination, the chromosomes line up, and so the hotspots are where all the repetitive elements are. If they are kind of close to each other, or even when they're apart, when they're so similar in sequence that sometimes they misalign. And that's where the hotspots are. And actually, all the microdeletions areas are full of them. And so, in fact, when we look at families, we have a lot of trios, and we can see that the kids always have something going on at those hotspots.
All right, and next question we have, audience member says, it's known that in hematological conditions, it's very important to karyotype analysis. In such conditions, acquired, may the optical mapping be useful?
Yeah, so blood is so accessible. Optical mapping would be great for any kind of blood type disorders. And as I showed you in the presentation, BioNano can do a really good job in making karyotyping more specific and much higher resolution. Because when you do the G banding and so forth, unless you use kind of fiberfish and see a better resolution, the standard karyotyping will miss cases like the last one that I showed in the Cornelia de Lange syndrome, where you have a 3 megabase piece and fish is just hard to figure that out.
All right, and sort of an add-on to that question, Chris from Baylor College of Medicine would like to know if could the larger events have been caught if karyotyping was done originally?
Yes, obviously it will be, but the indication for karyotyping right now is still kind of limited, right? And as I said, for the case at Cornelia de Lange, which I think Baylor just had a paper out in May, of something very similar, many translocations. And so for that, karyotyping will catch all the main events, but the small events usually missed.
Okay. And can you combine optical mapping with CRISPR technique to fix DNA abnormalities?
Wow, fixing is going to be tough. But yeah, I mean, you can use optical mapping to figure out where things are, and then you can try to fix it with CRISPR. But I think the technology is still quite a bit away. Just on a side note about CRISPR, don't quote me, but we actually have seen off-target events by optical mapping that short read sequencing think that everything is fine, but when we looked at the examples, after CRISPR editing, that there are structural variation events off target. So that study has not been published yet, but I think you'll see that over time, because when you're only looking with short read, you're not seeing the whole picture for the genome.
Yeah, I think that's a pretty good point. That we've seen some of that data starting to creep up. But so next question, our audience member asks, several of the de novo examples you show occurred in regions that have one or more genes. Why did whole exome sequencing not pick those up?
That's a really good question. I think mostly because we give you examples where optical mapping has an advantage. It's really where you have insertions. Okay, deletions are fine. I mean, short read, you can find deletions pretty easily. But for insertions, short read sequencing is still kind of struggling with that, especially for the larger ones. When you see kind of higher coverage, read depth for those regions, you're just never sure. So I think that's probably the reason. Once you know that it's there by, optical mapping or by 10x genomic sequencing, then you can go back and look for the coverage, and you always see it. So I guess it's maybe when the short read analysis for structural variations come in better in the future, that may solve some of the problems.
Okay, and we have one from Joan at Kaiser University who asked, although you identified the changes in the patients you presented, how do you know that there isn't some underlying change or changes in one of the parents' genome that would lead to additional chromosome changes in sperm or egg, so causing defects in subsequent children?
That's a really good question. We actually have been looking into that, and so far we have seen no pattern. we definitely keep that in mind and see whether there are reasons why these kids have the structural variation changes. And so far we haven't seen the smoking gun. We have looked at probably, we probably looked at close to 100 trios now, and we really haven't seen anything that can help us, except for the fact that these phases are full of repeats. And so maybe for most of us, when that happens in our germs and in our germ cells, the embryos were not viable, so we never know. But some embryos survived, and that's where we see it. So so that's an open question. It's a really important question that hopefully we can answer in the future.
All right, thanks. Hayek at the Children's National Hospital would like to know, what is your experience with combined analysis of OGM and LRS? What percentage of SVs are called with both and individually? And then lastly, does LRS increase the breakpoint resolution of OGM, and how useful is that?
Yeah, so I think this is similar to the very first question I answered. Combining long read sequencing with optical mapping, I think to me that's going to be the standard of care in the future. And, as I said, it's hard to pin down the numbers because we're not doing all comers. When we do these studies, we kind of favor the cases where short resequencing do not give us any answers. So as I said, long resequencing is really based on what is the read length overall that you can achieve. So right now, for PacBio or Nanopore, you can do pretty long reads, but then once you get into the repeat regions, then that becomes a problem. And so, so I would say for anything that is less than, say, half a megabase, optical mapping can detect all the structural variations. And then for long read sequencing, really depends on the read length. I would say anything less than 100 kb, we can detect pretty well. But most confidently, anything less than 50 kb.
All right, and we have a next question, this sort of forward-looking future crystal ball kind of question. When do you think the combination of sequencing and optical mapping can be introduced in the clinic, and what do you think needs to happen to make that possible?
Yeah, so I think right now, basically, there are two obstacles. One is that the technology is still not widely adopted because it requires a lot of skill, both long read sequencing and optical mapping. You need a pretty experienced technician to make it work. So that's kind of a technical problem. And then the other, of course, is cost. But personally, I think if we just not do things one step at a time, like you do microarray and then short read sequencing and then karyotyping and so forth, and just do it once with this combination, actually the cost should be comparable. So I guess if we have good reference labs that kind of invest in both of these platforms and use it together, I think that can happen. So I would say, a really good skilled lab and reasonable cost, that should work.
All right, and we have time for one last question. One audience member would like to know, how well do you think this technology is at detecting things like loss of heterozygosity, UPD, and triploidy?
Loss of heterozygosity is really easy because with long read sequencing and with BioNano optical mapping, you can just see runs of homozygosity pretty easily. Triploidy, it's a little bit different because if every chromosome, you have 3 copies of every chromosome, then it's hard to detect. But looking at one chromosome that is, with 3, like a trisomy 21, this should be a simple task.
All right, well, thank you very much. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on the Clinical OMICs website at clinicalomics.com for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward the link to any of your colleagues and friends who you think might be interested. I'd like to thank Pui again for his very informative presentation, and I'd like to thank you, the audience, for your attention and very thoughtful questions. And a very special thanks to BioNano Genomics for sponsoring this webinar. Hopefully we'll see you again another Clinical OMICs webinar in the near future. Goodbye for now. Everyone stay safe and healthy.